Thank you now, and good morning, everyone. My name is Chiara Cirelli, and I come from the University of Wisconsin in Madison. And my task today is to tell you about the arousal systems and how, in different ways, they connect to consciousness. Now, historically, and even now, when we talk about arousal systems and we study them, we do so in the context of sleep-wake regulation. So not so much in terms of consciousness research, but in terms of sleep research. And what you see here is the definition overall of what arousal systems are. They are specific cell groups in the brain that are important either to initiate and or to maintain the wake state. And so when, as you will see, these systems are quieter or they have less activity, then is when sleep is allowed. Now, I want to stress this because there will be a major topic in this lecture, that sleep is a state in which we are partially disconnected from the external world. So the fundamental definition, the unifying feature that defines sleep relative to any other behavioral state, from anesthesia to coma, is partial sensory motor disconnection that is really very nicely captured in this painting by Rousseau. So by definition, when we are asleep, we are less able to react promptly to a stimulus that potentially could be very dangerous. So for sleep researchers like me, this is not only, an operational definition, but it also suggests very strongly that sleep does something very important. Because all animal species that have been studied so far do sleep. And as you know, perhaps not this week, but on average, we spend a third of our time and life asleep. So there must be a reason why we are in this very, potentially dangerous state for so long, and everybody does that. If the same kind of function could be done in wake, evolution would have found a way to find that. So, but that's another lecture. That's not what I'm going to tell you about. But you have to keep in mind really, so disconnection, partial disconnection, is what defines sleep. Just so that everybody is on the same page, here is how we distinguish the two major phases. We talk about two stages of sleep in mammals in general, in humans. One is non-REM sleep and the other is REM sleep. REM is around 20% of total sleep. And here you see, if you record from the scalp, but it will be the same if you were going deep with a deep electrode recording the local field potential from the cortex, you would see that the activity is like this in wake. So very fast activity, very small amplitude signal. And then in non-REM sleep, early on, we have these large and global slow waves. Then later on, in non-REM sleep, the waves are still there. The slow waves are just smaller and more local. Then we have this REM sleep, which has an activity, a brain activity overall, dominated by, again, fast frequencies, small amplitude. So it looks quite similar to wake. And that's why it's also called paradoxical sleep. Paradoxical because the subject is asleep, is unresponsive to some extent. But the brain activity, if you were to just look, is very much wake-like. And in fact, I challenge any of you, myself, if you were going to be shown simply that trace, you wouldn't really be able for certain to say this is sleep, REM sleep, or wake. That's why it's called, in the French tradition, it's called paradoxical sleep. But there are other ways, of course, to distinguish this. So if you record the eye movements, in REM sleep, there are very specific rapid eye movements. That's where the name, one of the names of this stage comes from. And the other is muscle activity. Muscle activity is high overall in wake, lower in non-REM, and very low, the lowest in REM sleep. Okay, so that's the basic distinction, wake, non-REM, REM sleep. Now, this is how our sleep, at least in normal conditions, not now probably, looks like. So we go through cycles in a normal 7-hour night of sleep. And so we cycle from wake first into this deep, slow wave sleep, this early non-REM sleep, stage so-called 3 and 4, where there are these very large slow waves. Then we go into the first REM cycle, usually around 90 minutes is one cycle in humans. And then we keep cycling 3-4 times in a normal night. But as you see here, there is a period early on in which there are all these big slow waves and the deepest non-REM sleep. And REM sleep instead is much more frequent late at night and in the early morning. Now, all this is sleep. So both non-REM and REM share the key feature that is partial disconnection. But they differ very much in terms of experience and conscious state. Because early on in this deep slow wave sleep is when almost all times, if you wake up a subject using a paradigm that in the lab with Julia have been using very often, that is called the repeated awaking paradigm, you wake up subjects from this deep stage, almost always they will say, I came out of nowhere, I have no experience. Instead, if you go here in REM sleep, it's almost always the opposite, that they will report that there was something, so that they were dreaming. Okay, so in terms of disconnection, non-REM and REM look alike, they are the same, but in terms of conscious experience, they are very different. And what I'm going to tell you about is that when we think about these arousal systems, there are kind of two flavors, two types of arousal systems. And the way now we know they behave across the sleep-wake cycle is for one type more connected to disconnection, the key feature of sleep. But on the other type instead is perhaps related more to having conscious experiences during sleep. So going back to this, arousal systems, again, that's what I just told you about. Now ask me to give just a very brief overview that I think is useful for students also from an historical perspective. I was trained as an electrophysiologist, and I started my career in medical school recording sleeping cats, because cats were a very popular model, of course, for sleep, because you probably know they sleep more or less all the time, and it doesn't matter day and night. We sleep at night, rodents sleep during the day, mostly during the day, so you have to adapt. Cats, it doesn't matter. So, I was trained as an electrophysiologist, and I used to record few electrodes, few neurons in the brain at any given time, until very recently. And electrophysiology has historically been considered, it's kind of was the golden era of electrophysiology, as we were saying with Alex, was maybe in the 60s, the 70s. So it felt like an old-fashioned method. But there is a renaissance in electrophysiology that is driven by this new kind of electrodes that are becoming available, the neuropixels electrodes. They are very high density electrodes that allow you to record not just few dozen, but hundreds of thousands of neurons simultaneously. And this has been happening over the last few years. And is actually one of the few examples that I'm aware of in which the technology was actually optimized and applied first and even published first in humans and then in animals, because they have been used, they are used extensively to probe the human brain for clinical reasons in epileptic patients to find the source of the seizures. So that's what many people in the field now are using. What you see here is one of Graham's subjects. You can implant these, we implant them in rodents, in mice and rats. What you see here is the trace of one of these electrodes. These neuropixels are laminar silicon probes, very long. So literally in a rat brain, they can span the entire brain. So what you see here is a coronal section of the rat brain. So from cortex to hippocampus, thalamus, hypothalamus, you can pick neurons across all these regions simultaneously, which was not possible before. And
you guys can, if you're interested, you can also ask later. I think Masami, you also do that, right? But the Alex are analyzing this data, which is already publicly available. So, you know, if you're interested, then, you know, ask these guys.
so these probes, so this is just the first generation, but there is an international consortium involved in developing and optimizing and these kind of tools. And so they're getting even better and more powerful every day. But this original type has more than 1,000 possible recording sites from which you can pick 384 channels. And again, from each channel, you can record not just one, but many neurons. And that's why you end up with many, many neurons. So in our lab, we do two types of experiments. In one kind that Joanna is involved here she's not here now, but in head-fixed mice, we can implant up to 6 of them. Or in the experiments that Graham does, these are chronic recordings, so we can record for 2 days continuously, 48 hours, in freely moving rats. So it's a lot of data, quite overwhelming, I must say, but it's also, even for, you know, an electrophysiologist like me, really remarkable, because it is as if finding again or discovering so many new things that supposedly you have been doing for your entire career. So it's overwhelming because, again, rather than having, you know, like here, just one LFP, one trace, now suddenly you have here, that's the span of the probe, 384 of these local field potentials. And here again is in that rat, you can see it simultaneously, that is so powerful, the cortex, the hippocampus, and the thalamus. And this is just an example across the 3 major states that I mentioned before. So you see, this is really the theta activity in wake. This is a rodent. In rodents, unlike in humans, because the hippocampus is quite close to the cortex and the cortex is relatively thin, it's very easy to pick up, especially from central and posterior cortex, the theta activity that is happening when the animal is awake and exploring, or very prominently in REM sleep. In humans, there is theta. We know that from some very few intracranial recordings, but of course in normal recordings it's much more difficult to see that because our cortex is thicker, the hippocampus is not that close. And you see in non-REM, for instance, not just this is a slow wave followed by a spindle in non-REM, but simultaneously here in the hippocampus, you can see this other prominent rhythm that is the sharp wave ripple. And again, it's just beautiful to be able to see them simultaneously. And of course, yeah, Alex.
So it looks like in REM that it's very similar to awake, is exactly as you said, except for the thalamus has some rhythmic structures that.
Yes, that we are studying. We are discovering new things. Yep. Yeah, and it depends on the kind of thalamus. And again, here we have the ability to look at, high order, low and primary thalamus, and it doesn't look like they behave in the same way. So for instance, one thing that I would just mention is that we have known for a long time that to go to REM sleep, you enter first a transitional state in which there are a lot of spindles, and then you go into REM sleep. But it turns out that in the thalamus, there is a huge amount of spindling even in REM sleep, which you can only really detect with this kind of method. Yep.
I just had a clarification question. So this is aggregation of 300 and something neurons, or what am I seeing?
These are local field potentials. So
Okay.
you are recording the postsynaptic activity.
Oh,
correct.
Oh, so like a.
So this would be like an EEG.
Mhm.
It's just that it's deep.
Right.
But local field activity,
So you don't know like which exact neurons?
we don't know,
Right,
except
right.
that
Okay, yeah.
you extract from the same probe,
Right, right.
then you can extract
Done.
the
Thank you.
units. And then from that, you can do spike sorting and figure out whether it's one neuron, many neurons, and that's the power. And so that's why here now, rather than having, maybe 10 neurons when you were lucky, then you have all these neurons, because each row here again is one channel, so one LFP trace, and it can be one neuron or several neurons. So this is already spike sorted. And again, you see here, just is the same recording here to make the point in non-REM, the slow waves that I've shown you are due to the fact the neurons more or less tend to do the same thing. Either they're spiking quite at high frequency, the same as in wake, but then they stop firing, they go off for half a second. And then they resume firing and they stop. They resume firing and they stop. Because they do so more or less simultaneously, at least in early sleep. But that's why you can sum up their activity, and you see even on the scalp, these large slow waves, okay? REM sleep looks much more like wake. And in fact, you see here, the firing is pretty much tonic and is massive. So that's another way what we say is paradoxical. The early idea that we had was that, well, when we go to sleep, the brain rests, everything gets quiet. It's not quiet at all. It's not quiet even in non-REM sleep. It's just that because if you sum up overall the firing, it's only reduced overall relative to this by maybe 15%. So it's not a huge decrease in overall activity. But definitely in REM sleep, if anything, overall is higher than in wake. So in sleep, neurons fire in a different way, in a different neuromodulatory environment, as I'm going to tell you, but it's not that they, rest in terms of activity. Was somebody? Yeah.
It also seems that in REM sleep, they also are synchronously slowing down, like stopping, but the duration of the firing is like larger than non-REM.
Yeah, this is just one example, but we do see, and we see in fact that there are some slow waves even in REM sleep. So I wouldn't take these as, representative, and it varies also across regions, but definitely, yes. Another thing that, we have known, but it's very obvious here, is the hippocampus. You see here, this green especially, is actually more active in sleep than in wake. No, it's not yet been hopefully soon. So I just want to give you one example of how having this new technology is helping us to address specific questions. So, for instance, I mentioned in the discussion yesterday with some of you that we discovered a few years ago a phenomenon that we called local sleep in wake. So probably several of you, certainly myself right now, are kind of chronically sleep restricted or really acutely sleep deprived. So we are here, we are awake, we are behaving, hopefully performing okay. But the reality is that some of our neurons most likely are in a sleep state. And by that, I mean that they're going off exactly as they do here in bona fide non-REM sleep. It's just that there, more or less, they all do the same. They all go off. But in this phenomenon, that's why we call it local sleep in wake, is only a few neurons, a few small groups of neurons that do that at any given time. So the observation that we had, and it was first done in rats, and then we found it in mice, and then we found it in humans, is exactly that. So if we sleep deprive the rat, which we do in the lab many, many times. This is not severe, extreme sleep deprivation. This is just 5-6 hours of sleep deprivation. But if you record, at that time we didn't have neuropixels, so this is regular recordings. In early wake, so after just 1 hour of sleep deprivation, the LFP or the scalp EG shows nice activated EG. If you have here just 6 neurons, they more or less, they are firing tonically, like in wake, as expected. But then after 4 hours of sleep deprivation, sometimes you pick up in the local field potential this little wave, which is a theta wave. It's not as slow as a slow wave. It's in the 5-6 range, hertz. And corresponding to that, you see that this group of neurons, 6 of them, are off for just 100 milliseconds or so, all simultaneously. And then they resume firing. So in real sleep, non-REM sleep, if you have an electrode in the frontal part of the brain, in the posterior parietal cortex, you see that these neurons again are going off altogether, on, off altogether. But in this phenomenon, while the animal is awake, is local, because if you have 2 electrodes, you see they are now off here, but not in parietal. And then a few seconds later, they can switch. Okay, and what we found that is shown here is that the problem gets worse the longer you stay awake. And so the number of these off, that seemingly seem unpredictable, start coming up. And again, I'm sure in my brain there are many of them right now, okay? Now, we found these in mice, we found them in humans. So, and in fact, we have a large set of experiments now in both humans and animals to try to understand how much of what we know is the cognitive impairment due to sleep deprivation is actually due to this phenomenon, which again is subtle, because again, if you look, I look awake, I hope, correct? I can move around, I can jump probably, but again, this is happening. And you can show, in this study, we show that actually, if this event happens, let's say in motor cortex, just a few hundred milliseconds before the rat is asked to reach for a sugar pellet, it does not, is not able to, the performance is affected, of course. So there are negative consequences of this phenomenon. Yep.
Quick question. Is when rats are moving or not moving, does it matter?
Also, yeah, also when they're moving. I mean, moving helps, and I'll show you why it helps, because most, if not all, the arousal systems are engaged the most by locomotion. So that helps, but still in prolonged sleep deprivation, that's not enough to overcome sleep pressure and to avoid this phenomenon.
Moving, but the local is still sleeping.
Yes.
Wow.
Yes.
zombie.
I don't know if, but my experience when I was a student, a mechanical student, I mean, it would help studying by moving, you know. That's the best trick that most people can use to be awake. So, yes.
I'm not really sure of what you mean by some cortical neurons going offline. My understanding is that even if during sleep, the neurons are activating, but for example, on non-REM sleep, the neurons are in simultaneous activity. So that's why the EEG activity we measure is the amplitude itself is slow.
Yeah, that's exactly what I try to say here. So
Right.
neurons are either on or off. Off meaning they stop firing. And because they do that, then you see these slow waves. But if here it was only, you know, very few of these neurons doing that, you would not pick up this from the scalp EEG. And that's why this local phenomenon was not discovered for a long time.
So it's not like the neurons are totally inactive.
Well, they are totally not firing for 100 milliseconds.
Oh, I
Okay,
see.
so see, this is 200 milliseconds. So they are off here for 100, 200 milliseconds, which is the average duration of the off also in sleep. But then they resume firing, and it's a very small group of neurons.
Thank you for the clarification.
Sure. Yeah.
Quick related question. So these off periods are related to the slow waves. Are the slow waves traveling waves across cortex?
For this, we don't know. We will know soon, thanks to the neuropixels in which we can have a broader. But you're right, in normal sleep, the slow waves can travel, so that these off periods happen somehow in an ordered way.
Because in the neuropixel data, it looked like the hippocampus and the thalamus were synchronous with cortex.
Yeah, on that side, but you know, that's just one probe. So if you were to probe different cortical areas, I don't know what we would find. That's what we are trying to. And that's exactly, so here in this study, we had basically 2, at most 3 electrodes. So we call it local, meaning it could be in frontal, but not in parietal and occipital. But that was it. And the number of you know, neurons that we could record really was very small. But now, this is an example in one of these rats that has a neuropixel going across many different cortical areas from, you know, the frontal, this is the medial prefrontal cortex in a rat from M1 or cingulate to prelimbic, infralimbic, up to the orbital cortex. And that's what you see, and that's here, you can detect these off, and you can measure not just how long they last, but also the span, so the spatial extension that varies. And basically what we are finding is that indeed, also in terms of the number of neurons that are involving, increases with sleep deprivation. And we are trying to figure out whether there are some brain areas that are more susceptible of these than other cortical areas or even subcortical areas. So that's just to give you an example of how we can use now this new technology to characterize much better this phenomenon. And again, I don't know if you know of this. This is the most, the famous Randy Gardner. This is the world record of sleep deprivation. It's not a real experiment. He did it with a group of friends in San Diego to beat the world records of being awake for, he succeeded in beating the record. So he stayed awake almost 11 days. And that's how we looked like at the end of the experiment. Until the very end, until the last day, nobody knew about this. Only the last 2 days, and then for a week later, he was contacted and studied by a group of sleep researchers in Southern California. But again, it's not a real experiment. But in any case, what he was reporting, of course, was difficulty in being awake, sleepiness, hallucinations, actually. But definitely in a person like him, no doubt that there would be many off periods in wake. And we think, again, that a lot of the cognitive impairment that is subtle and is not when you are so sleep deprived that you really crash. You have what we call micro sleeps. Micro sleeps is when you lose tone, control, and you are really asleep for a few seconds. That's real sleep, just short. This is before. But it's definitely, we have a lot of evidence now in humans, it's definitely responsible for a lot of the cognitive impairment. Yep.
I was wondering, what do you think these off periods, how they're related to narcolepsy? Like, do you think that there could be some mechanism where these local kind of off periods don't propagate, or they suddenly propagate? So
I will get to that because I will talk about the orexin system. I don't think they are directly related to that. Although it's true that the orexin neurons are very important to maintain long wake, but I don't think there is a direct link between narcolepsy per se and this phenomenon. Yep.
So I was wondering, because you said that microsleep is a normal sleep, but very short. So can we even see some kind of stages of sleep in this microsleep, or what kind of electrophysiological pattern you would think there is?
You would see, definitely you would see a kind of small slow waves. So what I call N2, stage 2. When you go in non-REM, there is a transitional state, then N2, and then N3 and 4 that now are just considered one state. So it's really sleep. It's just that it's short. But more or less all neurons would go off in this bistable mode.
Sorry, just so is it daydreaming, like REM sleep, but short?
Possibly. So there isn't enough data directly to compare the two conditions. But, that's one of the questions that we want to address. Okay. So now I'm back to what I'm supposed to tell you about today. That is the arousal systems. So I told you already about this. I'm just adding this here, which is the view of what really neurons are doing in this space. So when they wake, or in REM sleep, as we say, they are tonically active, so firing more or less all the time. This is a cortical neuron. These are classical recordings by Mircea Steriade in cats, but it would be the same in us or in mice or rats. So that's the non-REM or slow wave sleep, stage 3 and 4. Then they go up, so on or off, on or off. It's not only on and off, it's also up and down. And by that I mean, and that's what you can do here only through intracellular recordings, is that if you record the membrane potential, you see that the on, when they are firing, the membrane potential is depolarized. And when there is the silence, the off periods is also associated with hyperpolarization. Okay, and then on. So that's why if you do extracellular in the papers, if you do extracellular recordings, we should just call them on-off because we don't know. But intracellularly, we also know that it's up and down. And then again, in REM sleep, instead, everybody's firing, neurons are highly depolarized. So arousal systems, there are, should I? here. Yeah, but it doesn't. I don't see it. Yeah, that's why. Yeah, then I go out.
Maybe we
Ask. Perfect. Okay. Perfect. So there are many of them. And again, you may think that we have known this for a long time, but again, even for in this field, there are so many new tools that are available. So, and I'm referring specifically to optogenetic and chemogenetic tools that I will very briefly mention, so that now we know there are many of these arousal systems, and I'm sure we will keep discovering some of them. So it's still work in progress. But what you see here is several of them. This is a sagittal view of the cat brain, but again, it's the same in humans or rodents in general. And you see that depending on the region, they use a different neurotransmitter. So I'm going to tell you, pick a few examples of these arousal systems. So the main point for this audience that I want to make is that there are two types again, based on how they behave during sleep and wake. So one type is, I lost the control of my, one type is this, in which these neurons of the noradrenergic system, orexin system, histamine system, serotonin neurons, they are all firing a lot in wake and very little in both non-REM and REM. And instead, this group is very different because they are quieter in non-REM, but they're actually firing as much, if not more, in REM sleep than in wake. And so now you understand why it's likely that, and there is evidence, that this group is more connected to the fundamental feature of sleep, disconnection, which is shared across non-REM and REM. And I will show you that especially the noradrenergic system is the one that causally has been involved the most in being connected or disconnected. And instead, this system, intriguingly, perhaps is more related to having an experience, whether or not you are connected. Okay, so let's start with, I want to start with the noradrenergic system, but before that, this is the view now. There are many, they're different. This is the old view that we got using the old-fashioned classical electrical stimulation method that was available, the only one available really until very recently. And this is the seminal paper by Moruzzi and Magoon when Giuseppe Moruzzi was working in the US with Horace Magoon, and when they discovered what is called the activation, the reticular, ascending reticular activation system. Who is aware of this paper and the finding? Okay, good. So, this was in cats that were either anesthetized or specific lesion, or they were put asleep. And the fundamental finding was that these are 4 traces, recordings in the brain, in the cortex. So under anesthesia or sleep-like states, they would have these slow waves. But the moment, and this is the time of the electrical stimulation, the brain was electrically stimulated, then the EEG would become wake-like, desynchronized, high frequency, low amplitude. Then the moment the stimulation stopped, the slow waves would resume. And this system was very widespread. So any area here from the medulla oblongata to the pons to the midbrain up to the lower part of the hypothalamus, anywhere you were going to stimulate there, you would get this activating response. So it was very large system responsible for the waking activation, wake-like activity. Now, this is yes.
Why is it called reticular?
I'll get to that. So this paper, I urge everybody, it's really a seminal paper. It's actually called in EG clinical neurophysiology a short communication. It's actually probably 20, 30 pages. It's extremely extensive. You know, now it would be impossible to publish something like that. A lot of observations are there, which I'm not going to discuss, but they were already there in 49, some hint of this major finding that I told you about, that there is more than one system and the system is very robust and is very broad. For instance, even animal with atropine, so with total block of the cholinergic system, you would still be able to induce this activating response, meaning that, you know, the cholinergic system in itself is not necessary. Another very important observation was that even so, if you stimulate the middle of the thalamus, you can also induce this activating response. But even if you lesion the entire thalamus, you still get, by stimulating the brainstem, the response. So, you know, even without the thalamus, you can have this response. Now, these are coronal sections to show that this system is mainly located around the midline, so it's a paramedian system. And why reticular? Because reticulum means in Latin means mesh, and the neurons in the brainstem look like this. And when in the adult brain, they really form a mesh. So they are very highly interconnected to each other. And many of them are very large, so-called Golgi type 1 neurons, very large neurons with a lot of afferents reaching their dendrites and very broad axons. They can span and reach different brain regions. Okay, so that's why it's called reticular.
This like it's not like very different from the grid-like structure that Julio talked yesterday, or is it similar?
Oh, no. Well, I wouldn't. So again, this is the old view in which everything is put together. Now I'm going to show you that this is composed of subtypes, and each of them, they share common properties, but also each of them has some features. I wouldn't call them grids. That would be my answer, I mean, short answer. So let's talk about some of them. First of all, in general, and that's why I mentioned this issue about the lesion of the thalamus, overall, they, to get to the cortex where, you know, the final effect is you have this brain activation, e.g. wake-like activity, if you stimulate here, they can get there in two ways, major ways, either through the thalamus or skipping the thalamus, and that's why even if you kill the thalamus, it doesn't matter, going down through the hypothalamus, basal forebrain, and then the cortex. So system is very redundant. And it's redundant because, again, it's made up of different subsystems. I'm showing here only a few of them: noradrenaline, dopamine, serotonin, GABA. Also, in many areas now we know GABA is actually activating, is not deactivating, orexin, acetylcholine. A major feature of these systems is that most of them are connected to each other, and they tend to excite each other. So they are helping each other, in this case, to maintain the wake state. But there is redundancy, but there is also specificity. So if you go then and study these systems, they have unique features, like the noradrenergic system is very important for the response to novelty. Dopamine, of course, you know, is related to a reward. Orexin, that's where the name comes from. Oreo means to eat in Greek. So it's related to sleep and wake, but also to food consumption, for instance. So each of them has also other specific functions. The other general feature is that these systems, they excite each other, but then they are also connected to the sleep cell groups. So I'm not going to tell you about this, but there are also cell groups in the brain that are important instead to initiate and or maintain sleep. And contrary to what we thought maybe 30, 40 years ago, there isn't just one sleep center. There are many, again, connected to each other. And the wake centers tend to project and inhibit the sleep centers. And vice versa, the sleep centers, many of which are GABAergic, but again, not all of them, tend to inhibit the wake centers. So the system is very robust. You are either in a wake state, usually, or in a sleep state, because it will be very maladaptive to be half the way, which is what we are, again, with the local sleep when we are very sleep deprived. So going back to this point, again, I'm going to tell you first about just two examples of these systems that behave in this way. This would be their activity firing rate, which we think then are probably related to disconnection, and then about these two systems that are more related to the maybe having a conscious experience. So the noradrenergic system is located here. The locus coeruleus is in the dorsal part of the pons. Here you see another view. This again is a sagittal section of the cat brain. And here is the locus coeruleus. Coeruleus means bluish, because these neurons have, at least in some animal species, fluorescent color. And here you see a major feature of all these systems. That is that they are actually, and going back now to your question about the grids, they are a small group of neurons, each of them, usually compact in a small region, small site, but they have very broad projections. We used to think that the locus projected everywhere except the basal ganglia, and now this is no longer true, because now we have better, even to do neuroanatomy, we have better tracers, and through them, now we know that actually the locus also projects here to the basal ganglia. It's not a huge, very strong projection, but it's actually there. And here is the evidence based on unit activity. So you put an electrode in the locus coeruleus and look at how they fire across the different behavioral states. This is active wake, quiet wake when they're not moving. This is drowsy state, slow wave sleep, paradoxical sleep. And you see that's the profile that I mentioned before. And one important point that I want to make here, see, this is an example, this is 1 second, of a transition from paradoxical sleep to wake. This is the muscle. It's so low that actually you can pick up the heart rate. So what you see here, that's the heart rate, okay? It's not muscle activity. I mean, it's the heart. And this is the EEG. And here is the moment the animal wakes up. First, the brain wakes up and then muscle activity starts. This is the unit 1 LC neuron. And you see that actually it starts firing before the waking up. This is just the sum activity. So both the green trace here and the black, these are all LC neurons. They are divided here in two, but it doesn't matter for this discussion. The point here is that relative to the zero, that is when the brain wakes up, these neurons start picking up activity before. And that's why we think they are very important actually to initiate waking, not just to maintain waking, which is very different from these neurons that are histamine neurons. Those neurons are wake active, but actually they start being active only after you wake up. So, but that's, of course, that's what we have known for quite some time, but that's correlative. Yes, sir.
Good question. Do we know what is the time, what is the distance between brain waking up and first muscle activity? Because it was shown here, and I don't know.
It varies because it depends on the muscle, but it can be 1 second, it can be less than 1 second. It really varies. Okay, so that's correlation, correct? So really the way the causal experiment to show that this system is important to initiate wake is to activate the system very specifically, only those neurons, and see whether the animal, if it's asleep, wakes up. And that's basically what was done in this experiment using this optogenetic method. Who is aware of optogenetics here? Okay, so many. So this is a wonderful new set of tools that we have. These are specific, usually pumps, either carrying anions like chloride or positive ions, sodium or calcium. So when you open the channel, the pump, the ions go in, and they can either hyperpolarize and inhibit the neurons or depolarize, excite the neuron. And we can have, you can choose. Now there is really dozens or more of these kind of opsins. And the locus coeruleus was one of the first systems in which the in vivo experiments were used. And it's called optogenetics because the way to activate these pumps is through light. And the light is usually delivered through a laser fiber. And so many studies are using the excitatory opsins, so they want to drive the neurons and excite them. And the beauty again is that now, rather than like with the electrical stimulation a la Muruzzi Magoon, in which you're stimulating everything, cell bodies, fibers, you don't really know who is affected. Here, you can engineer a mouse to have only the LC neurons expressing this opsin. And what you find then is the following. This is a control mouse in which the LC cells are expressing these yellow fluorescent proteins. So they are yellow, but there is nothing, no other consequences, okay? And so the mouse here is in non-REM. You see the slow waves. The muscle is very low activity. You shine light for 5 seconds here, nothing happens. You continue to see the slow waves. But here instead, you have a mouse in which, yes, they're yellow, but they are also carrying this channel rhodopsin, excitatory opsin. So when you shine light now, immediately the EEG desynchronizes, it becomes wake-like, and the animal wakes up. You do the same in REM. This is the control, nothing happens. You do here in the channel rhodopsin mouse, then the animal wakes up. You'll notice that there isn't much of a difference in the EEG because the EEG is already activated, but the muscle increases. So the point here is just that this is super powerful system. As long as you stimulate frequently enough and long enough, the animal will always wake up if you stimulate the locus coeruleus within a few seconds, and it doesn't matter whether it is non-REM or REM. It's very, very powerful. This is acutely, but that doesn't mean that if you lose the locus coeruleus, you are not able to be awake. And so this is an example. This is a mouse that was engineered to be unable to produce noradrenaline because it's missing this dopamine beta hydroxylase that is the final enzyme necessary to produce noradrenaline from dopamine, okay? But these mice are born like that. And if you measure their overall amount of wake, non-REM, REM, relative to the control, there is no difference. Okay, so, you know, in chronic conditions, and we go back to this issue of redundancy, fortunately for us, you know, you need to lose many of these systems before you see an effect on the sleep-wake cycle. Now, you do see an effect in chronic conditions. That's a study that I did a long time ago when I was a student, actually. There was to lesion the locus, in this case, unilaterally. The locus, many of these systems, arousal systems, have another feature that is that they are bilateral. Their projection, most of their projections are ipsilateral. So the left locus projects to the left brain, 95%, and the right to the right. Okay, so if you lesion, here again, that's a coronal section of the rat brain, and the black area here is the locus coeruleus. And here you see in this animal, given a toxin locally, we have killed this side of the locus, while this side is spared. And then if you look in the brain, in the cortex, here the gray are all the noradrenergic fibers. On this side, there are no noradrenergic fibers in the cortex. Now, this shows that if you keep this animal awake, this animal is normal, has a normal amount of wake. The EEG is normal on both sides. But if you keep the animal awake for several hours, while normally in the cortex, there will be many genes related to plasticity and long-term potentiation activated because the animal and expressed because the animal is awake and exploring. In this animal, these genes like ARC and many others, BDNF, FOS, and GFIA, these genes are activated and expressed on this side, but not on this side. This side, although the entire animal, of course, is awake, this side looks from the plasticity-related genes perspective as if it were asleep. So the locus is important during wake also to mediate, you know, plasticity and synaptic potentiation. So there are consequences, but overall the animal is awake. And there is now evidence that really can be obtained only playing around with these optogenetic tools. Some of these opsins, excitatory opsins that I told you about, there is one type, so-called step function, that when activated by light, doesn't really drive neurons to fire, just subtly excites them just a little bit to increase their overall excitability, so that then are more able to respond if a stimulus comes in. So using this kind of tool, it was shown that if you shine light to the locus during sleep, but not much, so that the animal doesn't wake up, is still asleep, but then if you give a stimulus like a small sound, the animal is more likely to wake up. Okay, so it's a complicated study to describe, so that's why I'm just giving you here the reference, but that's the really direct evidence that this system is very important because it's normally shut off completely during sleep. Is very important to keep us disconnected from the environment. And instead, if you are a little bit with the system on, you are still asleep, but much more likely to wake up. Okay? So that's the summary. So it's sufficient to induce EEG and behavioral arousal. I told you the moment you stimulate, the animal always wakes up. But it's not necessary because there is redundancy. And again, the low activity is very important for sensory disconnections. Of all the systems, the arousal system that we know of, this is the one most causally related to sensory disconnection during sleep. And I think I'm going to briefly go through two other systems that behave like the locus, but they are less important, let's say. And then we can stop, and then I'm going to tell you about the other two systems. So histamine is just to say that on one hand, the histaminergic system that is located here in the hypothalamus, the posterior hypothalamus in the region called the tuberomammillary nucleus. Again, it's just a compact group of neurons all together with very widespread projections. But I mean, it's wake active and doesn't, fire in sleep like the locus. But the fundamental difference that I hinted at before is this, that they only get activated after you wake up. But they are very important for maintenance. And you probably know, if you take antihistamines, they have very powerful effect on inducing sleepiness. So related to the question about narcolepsy, so this system, though, can be also, I mean, at least we have some evidence that this system may be also important for us to be connected when we are awake. And what is the evidence? The evidence comes from this disorder that is narcolepsy. Now, narcolepsy is a human disorder that now we know is due to lack or deficiency in the orexin system, orexin neurons that I'm going to tell you about. But even before we knew that, we knew that there was an animal model. These kind of dogs have normal, I mean, they are narcoleptic. They have all the features of human narcolepsy with cataplexy. And so like humans with narcolepsy that are basically unable to stay in one behavioral state for long. So they tend to transition from sleep to wake or vice versa many, many times. And so on one hand, they are very sleepy and so they have hard time to stay awake during the day. The other hand, they also have hard time to sleep in a consolidated way during the night. But also most narcolepsy cases are associated with cataplexy. So people and dogs, if they get excited, and very often it's positive emotions, it's not negative emotions. If they start laughing with friends or you give these dogs chocolate, they will get to the floor and they have a cataplectic attack, which means they suddenly lose muscle tone. Of course, we cannot ask dogs, but in humans, narcoleptic subjects will tell you, I could not move, but I was there. I was connected. It's just that I could not move, which can be very frightening, of course, at the beginning. In dogs in which invasive recordings have been done, it has been shown that, here is the normal profile of histamine, serotonin. I'm not going to talk about them, but it's like noradrenaline cells, very high activity in wake, much lower in non-REM and REM. They all behave the same. But the last column here is during cataplexy. And during cataplexy, noradrenergic cells and also serotoninergic cells are not firing or firing very low, like in sleep. But histaminergic cells are firing a lot. And so we think, based on this evidence, that the fact that these animals are able to be connected during the cataplectic attacks is due to, at least partly, to the histamine system. And finally, I'm going to tell you about the orexin hypocretin system that is also located here in the lateral hypothalamus. So again, same features. They are all there. They've been discovered through independent search for neurons with specific genes very recently, I mean, 15 years ago. Small compact group with very large projections everywhere, including a very strong projections actually to the locus coeruleus. And in fact, now we know a lot of their effects are secondary to their ability to activate the locus coeruleus. So again, that's the profile. By now you know, so they shut off in sleep, both in slow wave sleep and REM sleep. And here is, this was actually 2007 was the first experiment in vivo in which optogenetic was applied in any field. And it was to prove that orexin cells are important for waking up. And so again, here is the same kind of layout, because these are actually the same authors of the locus coeruleus paper. So this is the control. You shine this again here now, this mouse has the activating opsin only in the orexin cells in the hypothalamus. And so when you shine light here, this is just a control and cherry, so the neurons are red, but nothing else. You shine light here, nothing happens, the animal stays in slow wave sleep, or here it stays in REM, and he wakes up only here after many, many seconds. But instead, if you have the activating opsin, he wakes up much earlier. Now, much earlier, but not as fast as for the locus. This system is not as powerful. And this, I won't describe this, but this is a very complex experiment that, you know, now we can do. You can have actually a mouse in which you can optogenetically manipulate 2 systems or even 3 systems simultaneously. So this was done to activate the orexin neurons, while at the same time inhibiting the locus coeruleus neurons. And the, you know, the effect was that, well, if you inhibit the locus, it doesn't matter how strongly you activate the orexin system, the animal will not wake up. So you need the locus. So this projection from the orexin to the locus cells is very important. And it was then first the system, the orexin system was discovered as a new arousal system. And then it was discovered that actually by these 2 independent groups that actually narcolepsy is due to a deficiency in this system, either really in the cells or in the receptors, in the gene that codes for the orexin peptide or in the receptor for this peptide. And by the way, in humans, usually, so in these models, in the dog and in mice, it's really usually a genetic disorder, so it's a mutation. In humans, it's almost never the case that is a mutation that drives narcolepsy, is that the cells get killed probably through an autoimmune response triggered by an infection, a flu usually, or streptococcus infection, something that happens early on in life and then triggers quite slowly, but not that slowly, the kill of all these cells. And within a few years, all of them are gone, and then you develop narcolepsy. Yep.
It's just a little comment that when you say that, I thought of the encephalitis lethargica, where people were very drowsy and sleepy, but they were conscious and connected to the environment. And it's also like, there's a hypothesis that it was caused by infection. And I was wondering, maybe that was the case with the orexin.
Yeah. In fact, well, people have looked back at that literature and von Economo and, you know, the cases to think that, well, probably some of those cases were actually due to this system that at the time, of course, was not. And you probably are aware that, you know, depending on where the lesion was, some cases were actually opposite. So the pandemic, that pandemic caused the encephalitis lethargica, but also in some cases insomnia, because if you kill instead the anterior hypothalamus, where many sleep centers are, then you have, you know, mainly wake state. So that's the summary here. And again, the major feature of based on the narcolepsy evidence is that system is important to stay in one state for long. But if you sum up all the little pieces of sleep and wake that the subjects have, the total amount of sleep and wake is the same. It's just that it's fragmented rather than being consolidated. Okay, so I think we should maybe take a break now, and then I can cover then these two systems that are maybe related to consciousness during sleep.
Thanks for your talk. This question is slightly removed from the core topic, but still related. And what you presented on is about systems which sort of manage wake and sleep. But I was wondering if you know if these systems are related to the sort of experience of sleepiness, like feeling sleepy.
Both. Yeah. So if you subjectively ask somebody, are you sleepy, are you tired? And then you measure how bad their performance is as a measure of tiredness, very often the two don't go together. There is quite a strong dissociation. So already to try to answer your question is quite difficult because of that. What we are trying to address is indeed the sleepiness or tiredness that we have also subjectively that must come from somewhere. Is it related to the fact that neurons literally are tired somehow or not? If it had been true that in sleep neurons don't fire or fire much, much less, it would have been probably easier to assume, well then, you know, if you stay awake, they really get tired because they cannot stop firing, and maybe that's, you know, the two go together. But it doesn't look like it's so simple because of what I've shown you. So we basically don't know.
Thank you very much. So I wanted to make sure I understood the local sleep phenomena you talked earlier about. So when you say like the animal in the state of local sleep, does it mean like, so there are multiple neurons in the areas are actually simultaneously going off and for the like a specific period of time, or like what's the kind of, not the definition exactly, but like.
Well, our definition was very operational, was, you know, we have these neurons. We have, at that time, we didn't have, let's say, more than 20 neurons, 10 in frontal cortex and 10 in parietal cortex. When the animal is normally asleep, they all together go off for 100 milliseconds, and then they come up. In wake here, only, you know, 5 in parietal cortex would go off. Everybody else not. But 5 going off simultaneously is enough that you see this little wave,
right?
And
True.
you see a impairment at the, you know, behavioral level. So what is the smallest number of neurons that are needed? We have no idea, and that's why we are trying to, you know, probe the brain more extensively with the neuropixel. Basically.
Thank you.
Okay, so now we are going to talk about the two other systems, the cholinergic and the dopaminergic, that have, as I mentioned before, a very different profile. They're very active in REM sleep. So about the cholinergic system, there are actually 2 groups in this case, not just one. So there is one system, here that is located in the dorsal pons, very close to the locus coeruleus. This system goes through the thalamus and then reaches the cortex. And the other group instead is located here in the basal forebrain. And from there, it projects directly to the cortex. Now, these systems talk to each other, and they have many different features. But overall, this system is more important for cortical desynchronization during wake and REM. This system is more related also to the control of REM sleep. So here is the profile, which was described. This is for the brainstem the basal, the brainstem cholinergic neurons. And again, it's very different from the locus, no? So it's active wake, quiet wake, slow wave sleep, transition to REM sleep, and then REM sleep. And again, you see here, they are even more active than in wake. And here is the other, the forebrain cholinergic neurons, but the profile is exactly the same. So again, active wake, quiet wake. So we go back to, if you're really sleep deprived, just keep moving. I won't be absolutely offended if you were to walk. I often, when I go to meetings and I have big jet lag, that's my way of dealing with it, just walking. Again, this is paradoxical sleep. In the French tradition, every paper published by French authors would call it paradoxical sleep, not REM sleep. That's a historical note. But we have known also, if you measure, it's not just if you record neurons, these classical studies in the 70s, that they were measuring the levels of acetylcholine directly from the surface of the cortex. And what they found was that the total amount of acetylcholine was double in paradoxical sleep and wake relative to slow wave sleep. So we have known for a long time that this system is active in REM sleep. And here is now find the, after the correlational studies, this is the causal experiment, one of them at least, in which you do the optogenetic stimulation. So again, here I'm showing you the basal forebrain because that's the one more directly related to cortical activation. And so these mice now are engineered so that the opsin is only expressed in cholinergic neurons. And here is the wild type, the control. So if you shine light here during slow wave sleep, the EEG remains with these big events that are slow waves. You don't see the wave here because this recording is very compressed. But basically the animal doesn't wake up. Instead, here is the mouse with the channel rhodopsin. And then when you shine light, the animal goes from slow wave sleep to wake, EEG activation, and the muscle increases. Now, this is another example from the same mouse. This time, you do the same, but actually, the animal, yes, the EEG activates here, but you see the muscle stays very low. So this time, the animal didn't transition to wake. It transitioned from non-REM to REM sleep, which makes sense. This system is active not just in wake, but also in REM sleep. And here is, this time, you do the experiment, the mouse is in REM sleep already. And if he's in REM sleep, he stays in REM sleep. And if he's in wake, he stays in wake. So basically, this system fights bistability, meaning when you activate the system will make sure the neurons are not on-off, bistable, like they are in non-REM, but they transition to tonic firing. And the tonic firing can be wake or can be REM sleep, but it's not necessarily wake. And if they are already in REM, they are very active. Even if you activate them, nothing changes. This is a complicated slide. I won't go into the details, but I just want to mention there is another tool that we can use. It's not optogenetic, but it's called chemogenetic. It's again a way to express a very specific, in this case, a receptor to the neurons that we want to study. These receptors are modified muscarinic cholinergic receptors. They are modified in a way that they actually don't respond to acetylcholine, but they respond to strange ligands that are not present in the normal subjects, in us or in a mouse. So you have to give the ligand. It's only when you want specifically to activate those receptors that then you give the drug, okay? So when you do so, then you see the effects. The difference is nice in the lab, we use them a lot because they tell you something different from optogenetic experiments. In optogenetics, the beauty is that the system is super fast. You are, either inhibiting or activating neurons within milliseconds. This system, being based on second messenger signaling, is much slower. So when you give the drug, then you see effects on whatever you are interested in our case, sleep, for hours. And so that's another way, a complementary way to study what is the effect of, in this case, manipulating the cholinergic system. And what I want to mention is just this, that if you give the drug and then you record the animals for hours, what you see is that overall there is this decrease, the blue is the effect, in the low frequency power spectrum. So in the brain, there are basically fewer and smaller slow waves, even when the animal is in slow wave sleep. Because again, this system, when you activate, it fights, it prevents the neurons to go on, off, on, off. So that's what I just said, opposes the bistability on-off that is typical of non-REM sleep. And so it helps the cortical system, the thalamocortical system, to switch to tonic mode. But again, the tonic mode could be wake or could be REM sleep. It's not necessarily arousal, waking, promoting. And that's what is here. Yep, yep.
And do we know what determines whether they go into wake or to REM sleep, or is it random?
No. So if they are, if from non-REM, you mean, probably in this experiment, it was not carefully studied to exactly to address your question. I would bet that depending on where exactly you stimulate in the non-REM period, because early on, as I mentioned, there is, non-REM is not a stable state. There is non-REM, and then there is this transitional state in which you start spindling a lot, and then there is REM. And so probably my guess would be that if you stimulate very early on, when the system is not ready to go to REM, you would wake up the animal. If it's more already in this transitional state, it's probably more likely that it would go to REM. But that's my bet. Yep.
So there was a notion for a while that if you are already alert, but you pay attention, then in a way the EEG gets even more desynchronized and that gamma power goes up.
Yeah.
And I think some people, at least in my mind, there was this idea, oh, acetylcholine just keeps on going up as well. But so that seems to be wrong then.
Well, to some extent, I'll show you. I mean, that a lot of that comes from studies with atropine or anticholinergic drugs. And to that extent, even if you are awake, you are performing and you have some levels of slow wave activity, the more you fight the cholinergic, the more you activate the cholinergic system, the more you have gamma-like, and the more you tend to inhibit the system within the same state, the more you have slow waves. So, it's probably still true, basically. Okay. So how does this happen at the molecular level? What does it mean to fight bistability? So the cholinergic system acts, as you know, through two major receptors, muscarinic receptors and nicotinic receptors. One major mechanism by which we go from the bistable on-off, up-down of slow wave sleep to the tonic firing of wake or REM sleep is through the muscarinic receptors acting by inhibiting leak potassium currents. So potassium currents in general help when they are activated to hyperpolarize, so to bring down the membrane potential and make neurons less excitable. Muscarinic activation blocks these tonic currents and makes neurons more excitable. This is a very general mechanism that is probably a major component of these effects. There are many others effects. The nicotinic receptors in general, many of them are presynaptic. So they are actually located on the axons of other neurons, and they are very often located on the axon of other arousal systems. So for instance, the noradrenergic neurons. And so when you activate nicotinic receptors, you are actually exciting, facilitating the release of noradrenaline from noradrenaline axons. So we are back to this system that, you know, the system is not only redundant, but the system help each other to try to, you know, get to the same effect. There is also this issue of wake systems fighting against the sleep systems. So another way is that actually nicotinic receptors are inhibiting directly some of the major sleep promoting neurons. The bottom line is that there is not a single mechanism. The cholinergic system has many ways of ensuring this transition. And in normal conditions, of course, is a mixture of both muscarinic and nicotinic actions. So going back to your question, there is this very interesting phenomenon that is still striking to see to me these days, that, you know, if you activate the cholinergic system, you see tonic activation and cortical firing. If you give a cholinergic antagonist, you can have an animal that moves around, looks awake, but if you record the EEG as huge slow waves. And this is QNB is a cholinergic antagonist. It can happen with scopolamine, it can happen with atropine, many cholinergic drugs. And there is a huge literature on this phenomenon of this dissociation apparently between behavior and EEG. So usually when we are awake, you know, the EEG is activated. So this old but very fascinating collection of papers is all about these anticholinergic drugs and the effects. And there are, you know, this has been studied in many, many animal models and in humans, and actually still very much topic of interest also for the military, because the anticholinergic drugs are used. And so people want to know basically how much a human being can perform despite being in an atropine state. So the bottom line of this is that the apparent dissociation is only apparent, meaning if you are under atropine, yes, you are moving around, but your performance is actually, you know, impaired. And it might not be so obvious if you don't use the right tests, but if you do, then it becomes apparent. But in a normal condition in a rat, if you give atropine, you can have, you know, in the rat in a cage, there is a limited amount of behavior that it can do. The animal is still able, for instance, to avoid hitting the wall of the cage. So it goes around, is able to, you know, move around. And so it's striking to see how, well, it looks, its eyes are open, it moves around, but the EEG is full of slow waves. Again, the moment the animal engages in strong physical activity, for instance, if he jumps on a running wheel or he runs a little bit more, then you see the EEG immediately activates. Okay, so, but this effect, it's very striking, and again, it's actually a topic of interest even now for the effects in humans.
If you ask people under this stage, then they all.
If I ask you now, what is your name? You will say, I'm here doing this. So wow. I mean, you have slow waves, but if I ask you to do a PVT, a psychomotor vigilance task, and I ask you to respond as quickly as possible when you see a stimulus, I will see that your, performance is impaired. But it's not so obvious that, you are there, but you are not there.
Consciousness-wise, unclear.
Consciousness-wise?
Phenomenally.
Yeah. What do you mean? Again, some level of behavior, of adaptive behavior is still there. Of course, it also depends on the dose, correct? But it's there to the point that, again, you can have atropine and still being able to operate a plane up to a certain extent. But if I test you, actually, it doesn't look like your performance is 100% for sure. Yep.
Just to be even more specific. So talking, for example, it's like some high level cognitive ability is still there.
Yeah.
It's just worse, significantly worse.
Yes. Yes. Yes.
And if you ask these people, do they feel normal, or do they just feel drunk or drowsy? Or
often they report hallucinations. And that's why I think it's intriguing. And again, this literature, this thing was very hot topic, so interesting in the 60s and the 70s. And that's where all this literature, was born. Now, to my knowledge, it's only in very specific fields, like in the military, that this is being studied, but not with your questions in mind. So it's something to revisit. But in the old literature, one thing that people report is that they actually have hallucinations, which is intriguing because that's the point that I want to make here, although, I don't know how to explain this, but it seems that there are these two opposite states. One is hypercholinergic state, which is REM sleep, in which basically you are always hallucinating, because that's the definition of dreaming. No? But also it seems that an anticholinergic state, atropine state, also leads to hallucinations. Why, how? I have really no idea. It's something for people who study consciousness to deal with.
quick question, could this be related to sleepwalking? Do we know what that state might be like?
No, it's not sleepwalking. Sleepwalking happens during non-REM as a different, no, it's definitely not related to that.
If you ask question about more logical related, how they react? Like if you ask a mathematical question.
I don't think they were ever asked. Okay. And again, basically it's a wonderful collection of this progress in brain research. It's actually available online. It's most of what we know, because a lot of these papers are in humans, not in, this has been studied in rabbits, in dogs, and the phenomenon is there in all species, but of course in humans.
About these different effects of acetylcholine, I just thought maybe there's this imbalance between like excitation and inhibition, that the homeostasis is not like achieved, and that's why we have this kind of like hallucinations or?
Yeah, it might very well be. And, you know, this is one experiment that could be done because, you know, in the same subject, you can study their kind of, you know, conscious experience during dreaming, and then you can treat the subject with some kind of anticholinergic drug. That would be still not impossible to obtain permission to do that in through the IRB and, you know. Okay. So the last system that I want to talk about is the dopamine system, which is also a system that has been ignored in the sleep field for a long time. And I'll tell you why. But the first neuromyth about the dopamine system is the following that I want to point out. So in most books that you find, probably what you learn is, or what you see is this kind of pictures in which the dopamine system, you know, there are two major groups. One is from the substantia nigra to the striatum, then the VTA in the midbrain projects to the prefrontal cortex. This is one book, this is another one, basically the same. So I'm not going to talk about the striatum and the substantia nigra. I will focus on the VTA, and that's how it's shown, okay? But then there is this kind of picture. And I would argue that this is actually a more faithful depiction of the dopaminergic system than this, simply meaning that it's not that there are only projections to the prefrontal cortex. The projection from the dopamine systems are quite widespread. And it's kind of, historically, it's interesting to understand why this happened. And I think if you read this review, this is a beautiful review that explains a little bit the history of this, of why it turns out that, you know, many people were interested in this connection and they get focused, and most of the field got focused on this and basically forgot about the other areas of the cortex that are also affected by dopamine. Okay, but it's interesting. And what is the evidence that actually dopamine is not just in prefrontal cortex? There are classical and anatomical studies in which this is in monkeys, in which they were, you know, cutting the cortex in different pieces and basically measuring the amount of dopamine in different areas. And just to give you a sense, this is how much noradrenaline in different areas, you know, prefrontal, front of the brain, back of the brain, occipital, temporal lobe. See, noradrenaline is almost everywhere, as I showed you, as everybody accepts. The only region that really is a little bit less is actually the occipital cortex. Okay, but that's noradrenaline. That's dopamine. Now, you see the same kind of profile. So it's not the case that only this part here has dopamine. Dopamine is everywhere, again, less in occipital, but it's not definitely not only in prefrontal cortex. So I just want you to remember this, because this is, to me, is fascinating how, you know, this kind of neuromyth. So that's, again, is the review that really explains historically why this may have happened. So why was dopamine so neglected for a long time? Because the firing, if you measure the mean firing rates of dopamine neurons across the sleep-wake cycle, that's what is shown here. It looks very boring, and it doesn't look like it's state-dependent. It doesn't change between sleep and wake. So it's very different from what I've shown you for all the other systems so far. And that's why sleep researchers basically were not interested. However, then it was realized, of course, that dopamine, most of dopamine neurons fire not just tonically, but also in bursts. And most of dopamine gets released when there are burst firing, not tonic firing. So functionally, the burst firing is very important. And if you look at burst firing, that is the dark areas here, then suddenly you realize that actually it is state dependent. So burst firing happens during wake in specific conditions when the animal is, for instance, given reward of chocolate or is eating, and it happens in paradoxical sleep, in REM sleep. So suddenly now we have a state-dependent profile. And so dopamine systems have been really only recently have become, again, under the attention of sleep researchers. But they're difficult to study because they're deep and because the cells are kind of scattered a little bit more than other systems. So one way many sleep researchers are dealing with that is by using fiber photometry. So rather than, recording from neurons, which is difficult because again, there are few and they are a little bit scattered, they are using this method in which they put a fiber inside the brain, deep where in the midbrain where the dopaminergic cells are, and they are recording the population activity of these neurons using, again, a trick. So using a mouse in which only the dopamine neurons are expressing a fluorescent marker. And the fluorescent marker is related, is linked to a calcium sensor. So basically, every time there is a lot of calcium inside the cell body, which is when neurons are firing a lot, then the green fluorescence increases, and you can use that as a proxy for firing. It's very easy to implement this method. Again, it doesn't give you cellular resolution, but it tells you that overall, the population of cells that have dopamine there are active or less active. When you do this, so what you measure here is not the action potential, the firing. What you measure is a delta F over F, so simply a change in the fluorescence across states. And you see overall this profile. So it's more in wake, least in non-REM, and again, the most actually in REM sleep. Okay, so that's one way to see, and then you can study how specifically these cells are changing during the transition, for instance, from non-REM to REM. You see that they start going up, and actually from REM to wake, they go down a little bit. So really the state in which they are the most active is REM sleep. And here is the causal experiment, because again, before it's just correlation, but now you have an animal in which these cells have the excitatory opsin. This is again the control. So if you shine light here for a few seconds, the animal is in no REM sleep, nothing happens. But the animal with the excitatory opsin wakes up from non-REM, the muscle increases. And this is happening both in non-REM and REM. This is the latency. If you stimulate at 25 Hertz, within 1 or 2 seconds, the animal wakes up. So it's very powerful from non-REM. From REM, it takes much longer, probably because these cells are actually already active in REM sleep. Okay. This is another way that this system is so, these tools are so nice because now people can not simply put the laser where the neurons, the cell bodies are, but actually stimulate only the terminals, the axons of these neurons. I told you the general idea of these systems is that they are very widespread projections. So if I stimulate the cell bodies, yeah, I can see that the animal wakes up, but if I want to know of all the places where the system is projecting, which one are the most important for waking up, then I can, stimulate only one site or a few sites rather than all of them acting on the axons. And that's what was done here. It was, targeting specifically the projections from the VTA to the nuclear accumbens or the prefrontal cortex, or the amygdala, or the dorsolateral striatum. Very strangely, surprisingly, all work to wake up the animal except the prefrontal cortex. So actually, when you stimulate dopaminergic cells, probably the waking up depends on the projection to the amygdala and the striatum, not to the prefrontal cortex, which is quite surprising. And I won't go into this. The dopaminergic system is tricky because it's really a system that we are still trying to understand. And we now know that dopaminergic cells not only are in the midbrain, but there are scattered dopaminergic cells also in the periaqueductal gray. So I don't know if I should go back. Sorry. So in addition to here, there are cells here, more dorsal, very, very close to where the locus coeruleus cells or the serotonin cells are. And again, they're difficult to study because it's not a compact group like the locus that I've shown you before. And so that's why people, again, are resorting to this fiber photometry. Take-home message, when they do so, they see the same kind of profile. So these cells are active or similar, if you wish. They're active in wake, the least active in REM, and active in the middle in REM. So these are more active in wake than REM. The other ones were more active in REM than in wake, but all are the least active in non-REM. This system is still very much under study. And there is already evidence that these are not the only dopaminergic cells that are there in the brainstem. It's just that they are scattered, so they're difficult to study. And this is just to show that, again, you can use the optogenetic method, stimulate specifically the cells in this region. And again, what you get is that you can wake up the animal both from non-REM and REM sleep. The point that I want to make here is the fact that we have known for a long time that the dopaminergic activity is linked to dreaming. And the reason is this kind of evidence. So on one hand, we know that for the dopaminergic system that is involved, that is affected in Parkinson, when people develop Parkinson and they develop dopaminergic deficits, many of them report that suddenly they stop dreaming, that they were having a lot of dreams before, but then they stop dreaming. And conversely, when then you treat them with L-DOPA therapy, they report that they resume dreaming. So there is a connection between dreaming and dopaminergic activity. And the second is this literature of lesion experiments that was collected mainly by Solms. There are 2 sites overall in the brain that he collected many cases where if you have the lesion, you lose the ability to dream. And these sites are located either here in the medial prefrontal cortex or in this parieto-temporal occipital junction. And so Solms believed strongly that, the reason why if you lesion this, you lose the ability to dream is because you impair the connection from the VTA, from the dopaminergic system to this. I just showed you that this connection is actually not important to wake up, but it might be very important instead for the experiencing of dreaming while you remain asleep.
About the ventromedial prefrontal part, so it is impairing the dream because it affects the passage of this dopamine system, but not really that area itself is important for dreaming.
It connects not the area per se,
Mm.
but the connections of the dopaminergic fibers, the
That go
dopaminergic
through it.
activity. Yes.
So
You know, these lesions, actually, most of these cases, these are often due to stroke or other kind of pathological lesions, e-collected. These ones were due to surgical lesions, leucotomy, performed to treat schizophrenia a long time ago, okay? But the end result is that you cut this connection. And because at that time, again, historically, there was this, not fixation, but basically this focus on dopaminergic system being just connected to prefrontal. The other evidence that we have is this. You know, that has nothing to do with the lesions or and that is quite, you know, consistent finding about dopamine and loss of dreaming. But the point that I want to make is that the dopaminergic system is perhaps the least understood. And it might be very well be that the connection with dreaming has to do also with other dopaminergic cells that we still really are very poorly characterized in the brainstem.
So the REM sleep EEG is preserved, but people stop dreaming. So could it be that they just don't remember the dreaming instead of not actually dreaming? So the EEG is compatible with wake EEG. Right?
Yes, yes, definitely.
And they just stop, but they report like not remembering any more dreams.
Yes, yes.
So could it be
they
that
report that, you know, these are especially patients who say, because many of us, I don't know if I ask around, but like me, I never remember my dreams. So I would not be a good subject, you know, to see this difference. But subjects who knew I was dreaming every night, I had fantastic, you know, engaging dreams, and then suddenly I lost this ability. We're talking about those subjects.
But maybe if you were to do like serial awakening, they would report. Could it be that they're just having a hard time remembering?
It could be, but
so I did a PubMed search right now with dopamine and consciousness, and it's true there is a sparsity of research on dopamine connected to consciousness. And besides these connections, are you aware of any more maybe specific hypotheses that can be made about dopamine and its role in modulating conscious experience, not necessarily only dreaming, like in awake consciousness? Some impairment of dopaminergic neurons or projections lead to specific deficits in conscious experience in the awake state.
No, I'm not aware of that, but it might very well be there. I don't know how, so basically the most available pool of subjects are those treated, you know, for Parkinson and then resuming. So I'm not aware of any study that follow them and ask them specifically about their wake experience or daydreaming or relative to dreaming and how they recover that. I don't know. No.
Just, if I may follow up quickly, from some personal experiments with minor dopaminergic neuromodulators like L-tyrosine, which has just a transient increase in dopamine, which the pharmacokinetics go pretty fast, so it's eliminated. I noticed as phenomenologically an increase in the refresh rate of the experience, especially if you couple it with some of more caffeine-derived noradrenaline. It seems that the flow of experience is a bit faster, and I can pack some sort of more information in a single unit of experience. And that's like, it seems like an acceleration rather than a slowing down, which I suspect in Parkinson happens precisely the other way around. Yeah, like an ossification of experience.
That's interesting.
Hello, this is a layman question. Let's say, thinking about the big picture here, like as humans, we need constant adaptation to our surroundings, as our everything is in constant change. I think this must happen at the neuronal level, and these neurons need adaptation. So what if that's why there is activation in sleep? It's a kind of let's say when we learn, it usually happens in sleep and when these neurons are not used. So let's say you cannot, let's say if you have a machine, you cannot beta test the machine when it's working. So you need kind of, what if during sleep, the neurons are doing some kind of beta testing for, let's say, adaptation. And then that's why we have sleep like a consciousness or dreaming. Consciousness is like a beta testing of the neurons responsible for consciousness. And here maybe role of dopamine, let's say, which has been, I think, associated with learning, could play a role. So this is just a layman's.
Well, the only thing that I can say is that the evidence that you can learn new material during sleep is not there. So you can learn very simple associations like odor and electroshock, these kind of, but nothing more than that. So I don't know when you say, or maybe I misunderstood, you learn during sleep. When you learning, one thing is consolidation of what you have learned during wake and refining that, that's a different story.
Yes,
but
maybe.
New learning, basically, you know, the whole literature, we all wish that you could learn during sleep and you study during sleep. That's not true. When it happens, is actually because you wake up and then you encode the new information. And the reason is exactly what I've told you for the entire morning, basically, that you need these arousal systems to be active to encode the new information. That's new learning. That's not memory consolidation.
Well, if you develop a system, or if you code an application, you need first the beta version of it, and then after this you publish the application. So I think it makes sense from a, let's say, neuronal level that if I was a neuron and I would make a, let's say, adaptation, then I would maybe first test that everything okay, Roger, and then let's go.
So REM sleep is brainstem sleep and is the ancient, ontogenetically, is the first of these sleep that we have. So when you're born, you basically have only almost all REM sleep. And people think the REM sleep is important because of all this firing to form connections. So if that is the beta testing that you are referring to, perhaps.
Yes, I don't have the correct terminology here. Maybe learning is something different. It's like when I practice a piano piece and then I next morning I came back to it, then it's like a different word.
Yeah.
Yeah. So.
Yeah.
I'm thinking about because dopamine plays also a role in curing, for example, ADHD, attention deficit disorder. I'm thinking now then for ADHD people, they have a more common situation of daydreaming. And this kind of daydreaming and this loss of dreaming and the attention and so on, is there some connection? I mean, chain them together?
Maybe, I don't know. There was a...
Hi. So dopaminergic drugs that promote wakefulness, like for example, amphetamines, do they do so through the dopamine or is it through some secondary mechanism?
Usually amphetamines are not just dopamine. So there are, you know, amines in general, including noradrenaline. So the they do promote arousal. I mean, they are still the most powerful. Let's put it this way: they are still the most powerful arousing drugs that we have. Although you can mimic, you can reach the same arousal levels by very high doses of caffeine. And caffeine works through a very different mechanism, by blocking basically the adenosine and inhibitory system.
Just a quick, dopamine is also super relevant for schizophrenia and hallucinations. So it's interesting that dreaming is kind of an hallucination. So
yeah, like for the cholinergic system, no, it seems surprising that you can get hallucinations both ways. It's probably a question of the balance. And of course, it depends, like for the cholinergic receptor, the D2 and D1 receptors are very different opposite effects. So it might be just that.
A follow up on Kenneth. I think from what I know, besides the de novo synthesis in the brain, dopamine is the precursor for noradrenaline. So you synthesize first dopamine, and then you can, through one or two more biosynthetic steps, you get noradrenaline. But then you can supplement with dopamine, and you have an increase in arousal energy, which is consistent with the idea that if you have more of a precursor, you have more of the byproduct, right? But then if you supplement strictly with noradrenaline, you have immediate increase in energy. So it seems that in your case, the dopaminergic drugs work mostly on noradrenaline to increase the energy levels rather than on dopamine.
Yeah, just a clarification on Nao's question. So from the VTA, is there like a bilateral projection to the medial prefrontal cortex and also all the way to the parietal, or is it more first to the medial prefrontal and then? I think I didn't get it.
Oh, so crossing, you mean?
Yeah, like if you so meaning, is there a direct projection from the VTA all across the cortex, right?
Yes.
It's not sequential.
No.
No, right?
No, so that was my point, that there are direct projections from the VTA, let's say, to parietal cortex. It's not through, from the prefrontal through cortical areas. The only region of the cortex that doesn't directly receive, it seems, is more the occipital primary visual cortex.
But then somehow the ventromedial prefrontal is important for dreaming and like initiating, or maybe in the network, or because it's related to like conscious experience. That's why I was thinking that it's not like the lesion directly doesn't mean that it's there, but somehow it's really important to kind of mediate this dopaminergic projection, the rest of the cortex.
So yeah, it might be that, or it might be that actually the effects of loss of the ability to dream due to those lesions has nothing to do with the loss of the prefrontal cortex per se. Is due to the loss of the fibers, of the dopamine fibers. The dopamine fibers, to get even to the posterior part, go through, those big bundles of fibers that pass through there. And that's why, these lesions, especially leucotomy lesions, they destroy all the connections. So it's not necessarily only the ones to the prefrontal. Now that we know that the cortex, and you might probably know the study that showed that what seems to be common to the having an experience during sleep, both in non-REM and REM, because we do have dreaming a lot also during non-REM sleep. What is common is a reduced amount of slow waves in the posterior cortex, in the region close to basically this temporal parieto-occipital junction. That's what is shared by any, conscious experience during sleep. they didn't find anything related to more frontal, medial prefrontal cortex.
So just to be super clear, so yeah, that arrow there should go all the way to the back.
Yes.
And the lesion just cuts there. So that's the point.
Yes.
And my question was before is like, how much inter-subject variability there is for this? Everyone is exactly the same, and
I will have to go back to the original study. You know, how much variability there is in this leucotomy?
in general for this mechanism, yeah. In general, I would say, yeah.
How much variability in reporting dreams or in what? Oh, in anatomy, probably not much. I mean, this system, this circuit is probably conserved. Yeah.
I want to go back to the point that you mentioned about how ACH antagonists affect. There was a dissociation between the behavior and the EEG activity.
Yep.
I wanna know why this happens, because I think this contradicts with the idea of reductionism. And if the EEG is dissociated with the behavior, I want to know what the EEG, where the EEG activity was measured, or what kind of EEG activity was desynchronized with the behavior, and yes, so I want to know the specification of the EEG activity.
Yeah, no, you're perfectly right. I mean, in those studies, you know, people in general, people put it maybe 2 or 3 leads. So when I show you the slow waves, they are probably frontal and parietal. That doesn't mean that other areas are not spared or have less slow waves. I'm not aware of any recent study in which now many of us use high density EEG, in which you could actually show that when you have a relative maintenance of performance or less impairment is because some areas are actually not so affected. And the basic knowledge also of exactly where in the cortex, which layers cholinergic fibers are reaching, how rich is the cholinergic innervation in humans, it's very difficult. It's basically unknown because we are still based on a few studies in animals. The best is in some primates, but it's very scattered information. It might very well be just depending on exactly where, which layer and which cortex, you have a very local, you know, picture.
I see. So there was a dissociation between a specific EEG activity recorded from a specific layer of a specific set of neurons, but it's not like there was a total dissociation between the whole brain activity and the behavior itself.
Yeah, at the beginning, people looked at this phenomenon as, wow, that's, you know, they have, think about the Moruzzi and Magoon that were coming from those kind of findings in which, you know, the two go together. Either you are awake and the EEG is activated, or you are anesthetized, there are slow waves. Now, suddenly you have this phenomenon that didn't make any sense. So they found this as, you know, dissociation. But as I mentioned, well, dissociation up to a certain point. When you test the behavior, and what do you mean by behavior? Just being moving? Yeah, it's possible.
I see. Thank you.
Any other question? Okay, well then, I think. Here's a question.
I think you brought it up already with this vivid hallucinations with the anticholinergic drugs. So are there any studies where they also try to block the 5-HT2A receptor?
not that I'm aware of, but I should mention related to this. So I told you about local sleep and the fact that, you know, if you're really sleep deprived, that you start having basically local slow waves. Local off means local slow waves. And although now it's basically impossible to do these experiments for ethical reasons, but starting from Randy Gardner, you know, world record, and then there are maybe 10, maybe maximum 15 papers published in humans severely sleep deprived. And most of these are studies done by the military in which you have permission. We wouldn't have permission to keep people awake for ethical reasons for 3, 4, 5 days. They do, or they did. Those very often after 48 hours, people start reporting hallucinations that they are very different from the classical psychotic hallucinations typical of schizophrenia. So they're usually not auditory, they are visual hallucinations. So, you know, it's intriguing that you have these in wake. At the beginning, when, you know, Randy Gardner reported this was, well, okay, fine, 11 days, it's one case. But based on the analysis, there is an interesting review about all these cases. It seems very consistent finding that after 40, 48 hours across subjects, you know, it's not hundreds, but it's dozens of subjects, hallucinations are happening.
Thank you very much for the interesting talk. In response to Miko's question, I think you already mentioned some developmental stuff. So like how, wait, what was the comment you made? Because I mean, about the REM sleep being like, you know, more like in infancy, like, you know, half of the sleep is REM sleep, if I remember correctly. And also like, I think until like mid-childhood or so, like a theta wave is definitely a lot more compared to like EEG wave wise. Theta and alpha flip, I think, around like 7 years of age or so. In terms of amount during the awake state. But in general, I guess like I was wondering if you can tell us anything about like these developmental changes happening either in the sleep state or about like these pathways.
So REM sleep, yeah, in newborns is certainly more than half. It can be, depends, you know, in preterms, it can be even 80% of all sleep. At that time, it's not called REM sleep, it's called active sleep, relative to the quiet sleep, which is the precursor of non-REM sleep. Now, one of the reasons for that is, if you think about it, is that operationally, the way we define non-REM, which I've shown you, is by recording slow waves, basically. Now, as the brainstem develops much earlier than the thalamocortical system. So when the thalamocortical system is still not developed, and we can study this as being studied in rodent models, until the first 2 weeks, P13, the first 2 weeks after birth in a mouse, the most cortical connections are not there. And so you don't record the EEG, the slow waves, I mean, or you record some activity in the cortex that is totally the same, independent of behavior. So the EEG doesn't help you at all because the substrate, you know, the synapses are basically not there. The first one to develop are the thalamocortical synapses already in the first week, but until the end of the second week, there is no strong cortical connection. And so those events on off, they just don't happen. So there, the only state, disconnected state that we have is the one in which is the brainstem state, which is REM sleep. Now then, you know, once you are able to define non-REM, that's at the end of the second week, which we are born already very mature. So in normal development, in new babies, you can already distinguish very well non-REM and REM. But then non-REM within the first year or so, the percent of non-REM quite increases, and the REM decreases. REM then remains quite stable in terms of percent. REM is 20% of our sleep and remains more or less the same across our lifespan.
You have like any comments also on that, how kids have slower, frequency during their awake time as well?
No, that I don't know much. I know it's very well studied because again, the EEG reflects cortical activity and reflects synaptic connections. So it's very well known that in kids, you have slow waves that are very large, that they peak in pre-adolescence, and then they actually, slow wave activity goes down, probably because of synaptic activity. And then by my age, for sure, but by age 35 or 40, basically most slow waves are gone. And so slow wave sleep, the deepest slow wave sleep is gone. Why is that? We don't really understand why. It's probably not because you are losing already all your synapses, but it's because neurons are less able to connect to each other to fire synchronously. Because again, to pick up these events from the scalp, you need many neurons to go on off at the same time. If they are still there, all the connections are there, but they're not exactly synchronous, then your signal will decrease. Or at least that's the optimistic explanation of why after 35 or 40, there is very little slow waves.
About this, so if the local sleep in a way is related to mind wandering, so that I and Thomas published a couple of years ago, you may know, which is also related to what Nata asked. And if you what you said right now, that older people don't have a slow wave, that it doesn't necessarily directly mean, but in older people, they do not also have much local slow wave in awake, and then less.
Yeah, no, that not necessarily. We don't know because we never studied, people across lifespan. But I wouldn't assume that absolutely. I'm talking about again slow waves, large slow waves depend on the synchronous activity. So local slow waves may very well be there even when you're 80, 90 years old.
And it may be actually it's increasing potential, right? Because
yes,
my
absolutely.
Is that older people tend to mind wander or maybe more often possibly.
Yeah, yes, it just has not been studied.
Okay, and then that's a small question, but I actually have a very kind of weird question potentially, but
good.
People talk about inverted Qualia and so on, right? I mean, in this summer school, my red Qualia, maybe your green Qualia or something like that. That's sort of a standard philosophical kind of question. Okay. And then, well, I've been thinking about this, all sorts of the activating system or cholinergic system and so on. That's a chemical-based stuff, right? And then most of the stuff that we have talked, all the way this week is at the level of the structures, at the level of either phenomenology or neural circuit or maybe the structure of synapses and so on. That could be related to sort of structure, but a chemical thing is also definitely a physical structural, thing, right? And then one observation that I always find very, very strange or fascinating is that very similar drugs are, have a very similar effects across almost all the animals. Mm-hmm. Like dopamine, dopamine seems to be doing something even from insects to octopus to here, and that evolutionary, it's almost very little is common among these species. So maybe it is dependent on this evolutionary history, but do you think that some particular neurotransmitter having this particular shape, and also that requires some particular receptor type, to act as a sort of key and lock, and then eventually that also dictates the probability of the opening or duration of the opening or something like that is almost like, required by the nature of the chemistry of physics.
I don't know. I only know that, among all these, perhaps I show you that perhaps the strongest evidence for the arousal is for especially noradrenaline. And noradrenaline is the one that is most conserved because in flies, noradrenaline is octopamine. It has the same strongest effect in terms of promoting arousal. Now at the chemical level, the receptor and, they're different and the brain is different, but the overall way the system is organized. So the fact that there is more than one wake group, more than one sleep group is conserved.
Yeah, but I always thought that, let's say dopamine can be can open the receptor that is, for example, potentially opened by the cholinergic, thing by doing some kind of artificial engineering of the receptors or something. Making this at the level of the receptor level, you create some kind of inverted animals. You know what I'm trying to say? Like, dopamine for us, it is kind of rewarding and arousing, but for some other animals.
Yeah, melatonin. Melatonin is an example that in birds, melatonin is actually wake promoting. And in us, it's sleep promoting. That's the example that I know of. But more, so for instance, we were talking about noradrenaline and dopamine. Now we know a lot of the effects, for instance, in the hippocampus that were ascribed from the locus coeruleus to noradrenaline are actually mediated by dopamine. And it's from the fibers, from the locus coeruleus, it's just that there is one step, synthetic step from dopamine to noradrenaline. And it seems that depending on the state or maybe the fatigue, you can release both or mainly dopamine or noradrenaline. But definitely now we know this is, it seems as if it was, at least for this effect in hippocampus, one system. So despite all what I told you, now the complexity is even that, the noradrenergic system is actually releasing dopamine, and you can block the effects of the locus coeruleus on learning by giving dopaminergic drugs, not noradrenergic drugs.
So the functions of the molecule coupling can be arbitrary, and one evidence is that melatonin system. But
yes,
roughly speaking, it seems conserved across.
Yeah, I wouldn't call it arbitrary because it's, after all, one is way, they are still affecting the same sleep-wake cycle, just in opposite way. I don't know, arbitrary to me means a little bit more like random. But
I have a follow-up on this train of bold ideas. Given at the level at which we have mentioned the concept of structure in the summer school and the way in which is used even in IIT, and maybe connected to something that Giulio said yesterday about there being a limited number of possible network configurations that yield sufficiently rich cause-effect structures, like an upper bound on possible experiences, let's call them like that. It might be that this type of morphological considerations for receptor architecture or even the drug chemical composition are ways in which you can, at an appropriate level of abstraction, configure sufficiently complex geometries for the networks that we are discussing, for brain networks, geometries that have consistently evolutionarily preserved motifs. And in this way, we can maybe give a sense of why we have a conservation of effect, even though they sometimes might be reversed, like in the case of melatonin in birds, but still the same principle holds. You have some little chemicals that you put them somewhere and then the network responds in a concerted way And that is seen across species, across different morphological organizations for brains, and so on, even evolutionary distant species.
Yeah.
So yeah,
Makes
like
sense.
it's more so of a unifying view of the
Yeah.
of things.
Yeah.
Yeah.
I am a bit intrigued by the enormous role of the subcortical structures in consciousness as you presented, as the evidence that you presented, and all the evidence also like you. So there is some evidence that layer 5 pyramidal tract neurons that project subcortically are more relevant or more important to perception, specific perception, than intratelocephalic neurons that are cortical, and regarding state and also content of consciousness, and also there's a nice paper that shows that that desynchrony in a layer 5 pyramidal tract neurons is what most correlates with waking up and going into anesthesia. So what do you think about that? So how do these subcortical nuclei desynchronize the cortex somehow, and what's going on in this dynamics between the cortex and subcortical structure?
That's a difficult question. My point here was that I see these systems are enabling the overall experience or not to happen. And so if they're all off, it's very unlikely that you have one experience, which would be in very early non-REM sleep. I wouldn't, ascribe to them directly much more than that. And then they are enabling when at least the cholinergic and the dopaminergic are very active. Probably that's, and so, I was, making it simple that, well, we have experience in wake and in REM almost all the time. In wake, let's say 100% of the time, in REM sleep, 95% of the time. Although it would be very interesting to be able to study that 5% of times in which people are in REM sleep, and yet when you wake them up, so supposedly they have a strong cholinergic dopaminergic activation, but then say, I was aware, I didn't experience. But the point is that the difference is not, the dopaminergic cholinergic system are active in both states. But between REM and wake, there's huge difference, for instance, in the noradrenergic system. So it might be that what counts is not just the absolute levels, but the balance between all these arousal systems together. And the fact that aminergic systems in general are very down is made as crucial as the fact that dopamine and cholinergic systems are up to provide the very specific conscious experience that you have in REM that we know very well. It's very different from what we have in wake. And so it's not just, they're sharing some activation of some systems, and yet they profoundly differ in terms of many other, and probably we don't know, there might be other differences that we don't even know about. And so going for these differences might be very informative to understand how the quality of the experience is actually, yes, they are both conscious states, obviously, but obviously nobody doubts that they are very different kind of quality and qualia. And that maybe that's the one way to go to try to understand. And again, that's something that at this level, you can still only do in humans. Asking very precisely about the report of the experience, and then trying ideally to measure the levels of these neuromodulatory. So I didn't mention, but it's possible there are sensors that are now available that, of course, are only used in epileptic patients, but deep electrodes that allow you not just to record like neuropixels, but also to measure different neurotransmitters. So the dream experiment would be in these kind of subjects to see how the balance, you know, in theory, it's becoming possible to do that.
I had a question more about what could be a possible function of REM sleep. And the follow-up question would be that in most of the cases, if experience is accompanied in REM sleep, what could be a possible explanation for animals having REM sleep?
I wouldn't rule out at all that animals do dream the way we do, actually. So I would start from that assumption. Of course, I will never be able probably to prove it, or at least not soon. But so, you know, I would assume that if they dream, and perhaps if there is a function of dreaming, is shared between us and animals in general. Now, you may be aware that we recently published a review on consciousness in sleep, in which we make the case that you know, sleep is a very useful window to understand consciousness, aspects of consciousness, but it's also the other way around. That through, you know, consciousness or dreaming, you can perhaps guess about the functions of sleep. And so the point that we made at the end of this review is actually that maybe the very strong activation, broad activation of neurons in REM sleep might be a way to basically reverse, activate all your possible memory traces and down select them. The idea that we have in general about sleep is that sleep is very important for synaptic downregulation. And that's something that we have evidence for across species, literally from flies to humans. Now, what we studied is always total sleep, of which 80% is non-REM. But it might very well be the REM is contributing to this downregulation of synaptic activity, but that in the way REM sleep does it, which is in humans basically dreaming, is by sampling in a comprehensive way all possible patterns of activity. Now, again, this is then something that is very difficult to test because animals can only help you up to a certain extent if you want to bridge with dreaming, but that's possible, you know, explanation that we elaborate on this review, but it's really, you know, just an hypothesis. We have no evidence for that.
This follows on Robert's question from earlier. I was reading this very interesting paper the other day that plants respond to anesthetics. So if you give an anesthetic to a Venus flytrap, it just stops working. And apparently they do have acetylcholine, but obviously the whole circuitry is very different. And so I'm very curious if you have any speculative thoughts on what kinds of mechanisms might be evolutionarily conserved.
No, Alex, we have been trying for several years to collaborate with these people. Some are in Italy, some are in Germany, that they report this very interesting phenomenon of anesthetics, volatile anesthetics, changing the reactivity of the root system. And so if that is the case, and there are these conserved mechanisms about sleep, because by definition, you know, the most physiological condition of unresponsiveness is actually sleep. And we haven't succeeded in convincing them to study, but I would not rule out that you can somehow have some measure of, you know, unresponsiveness in a behavioral state across the sleep-wake cycle, meaning across the 24-hour, without the need for anesthetics. I'm very open to that possibility. I just don't, I wish I could study that. It's interesting historically, you know, because the entire circadian field was born in plants actually, but yeah, 2 centuries ago, but people were looking at the leaves and how they react to the light, to the sun. While here, we are talking about reactivity in the root system. And these plant physiologists argue that what is the correspondence in plants of our brain is actually the root system, it's not the leaves. So it's fascinating.
Just a sort of announcement. So we will start the discussion report session from 3:30 as usual. But then at the end today, Chiara and Julia suggested to do the another talk on the free will at the end. Okay, so the discussion topic is about this stuff, but you know we might have things on the free will at the end.